Hi, everyone, and welcome to our symposium titled Emerging Research in Infant Neuroimaging. Infant neuroimaging is a rapidly emerging field. It's really have come to the forefront and matured over the past few years, and also with some NIH initiatives has really been gaining traction. So in this symposium, as the title suggests, we're going to be describing a few sets of emerging research that we have felt to be particularly interesting. You probably were wondering what the fitting portion of the title meant, or even how to pronounce that. That's short for an organization that we have founded called the Fetal Infant Toddler Neuroimaging Group. Due to infant and fetal and toddler neuroimaging becoming much more popular, yet probably not quite having a home, we've established this organization in 2018. And the goals of this are to provide a forum for early life neuroimagers to come together, build a community, help the field grow, and establish best practices. We have hosted a variety of events and plan to host a variety of events at various different conferences and in other venues, similar to this symposium. And if you're interested in fetal, infant, toddler imaging and want to get involved or interested in participating in these events, probably the best way right now to find us is using our Twitter. So here's our Twitter handle. Feel free to follow us, and we make various announcements through that. For the actual set of talks today, we have 4 speakers lined up to give a wide variety kind of overview of things. First, Silvia is going to be talking about kind of a survey of the literature over the past 20 years about a very fundamental issue in neuroimaging, registration, and what common space template we use for our analyses across various modalities and how that has changed over the years. Next, Alex and Lucille are going to be presenting kind of new work on the individual differences of both the connectome and resting state networks in infants. These are very hot topics in older individuals, children's, adults, but these are really the first time where we're getting to be able to use these methods in infant samples and really understand how this functional organization is established very early in life. And finally, Marisa is going to be talking about kind of more of an application of these approaches. So how do prenatal factors, specifically experience of discrimination in minority mothers, map on and possibly affect the connectivity of their offsprings in the neonatal period? So with that, again, I thank you all for joining us, and I hope this will be a very exciting symposium for you to listen to. Thank you.
Hello, everyone. My name is Silvia Gini, and today I'm excited to be presenting some findings from a systematic review of infant templates that me and my colleagues in the Minds Lab carried out over the past few months. Spatial normalization is an important image pre-processing step. It involves the registration of individual brain images onto a reference space, and this allows for the comparison between different groups and statistical analysis, despite differences in individuals' brain anatomy and morphology. In adults, the standard template used is the Montreal Neurological Institute one. However, for infants, no such a widespread and widely recognized analogous exists. And the MNI might not be best suited for infants' smaller brain and quickly changing morphology. So we were interested within this study to quantify what the most common approaches to spatial normalization in infant neuroimaging studies between the year of 2000 to 2020 were. And we also have a poster on this, so make sure to check that out too. So to answer this question, we conducted a PubMed search with the terms you can see on the slide, which resulted in around 37,000 initial papers. We then applied some exclusion criteria, including year of publication, language, type of study, age of participants, so they had to be scanned after birth and before 18 months, and also imaging modality. And we were left with a paper count of around 700. Within these 700 papers, we then went out to analyze what type of common space registration they were using, and we identified 4 broad categories. The first one was single subject space. So single subject space paper made no registration to a common space, but rather all the analyses were carried out into native space. These included examples of studies where a fractional anisotropy was compared within a specific region of interest. The second common space categories was study-specific templates, and these included the TBSS, for instance, and these were templates that were derived from the brain of the participants of the study itself. Third, we had off-the-shelf templates, which are templates that are predefined and have been validated before, such as the UNC or the MNI one for adults. And then we had a fourth category, which included the hybrid papers, and we classified papers as hybrid in two cases. The first one was if they had more than one common space registration within the same modality. So for instance, some papers registered to a study-specific template, and then the study-specific was registered to an off-the-shelf one. And then secondly, for papers that had more than one modality, if these papers had, each modality had a different common space registration. We then looked at how common each type of registration was in the initial 700 papers. And here you can appreciate the number of papers is slightly larger because different modalities were counted once for each different type of registration. And in particular, you can see that single subject space were the most common type of registration, followed by off-the-shelf, study-specific, and then lastly, there were also a handful of hybrid studies. The next question we had was what type of off-the-shelf atlases were most common in the infants literature and whether these would overlap with adult templates. As you can see here, the first and third most common atlases were actually pediatric atlases, so atlases that were created specifically from infants' brain images. However, the second most common one was the Montreal Neurological Institute one, which is an adult brain atlas, which is something we found quite striking and interesting. And the MNI actually made up around 14% of the total number of off-the-shelf registrations. The next question we had was whether the type of registration would vary within each modality, data acquisition modality. And as you can see here, some differences do emerge. For DTI, MRI, and MRS, the most common form of registration was in single subject space. Whereas for fMRI, the off-the-shelf registration or templates appeared to be most common. And notably, the DTI and fMRI had opposing pattern with single subject space being the least common in fMRI. We then wanted to assess whether different types of registration would vary depending on the age at scan. And as you can see here, first of all, the large majority of studies took place in the first 5 months of life, so from 37 postmenstrual weeks up until 5 months. And both this age bracket and the wide age range, which included samples that had an age range larger than 5 months and cross-sectional data at multiple time points, within these two age brackets or categories, single subject space appeared to be the most adopted type of registration, followed by off-the-shelf and study-specific. In all other categories, the numbers are quite similar, so they're comparable. However, in the longitudinal scans, there's a slight trend towards off-the-shelf being preferred over study-specific and single subject space. We then wanted to assess whether the choice of common space registration would also change depending on the participants recruited. We identified 3 broad categories. The first one was preterm babies, so babies that were scanned, were born before 37 gestational weeks, or low birth weight infants. The second one included other special populations, so both neurodevelopmental disorders and more medical conditions such as congenital heart disease. And the third one instead was no special population, so typically developing healthy children. And here I would like to draw your attention to the fact that clinical populations, so the first 2 groups, appear to prefer single subject space registrations over off-the-shelf followed by off-the-shelf and study-specific ones, whereas the no special population group appears to make more use of off-the-shelf registration. Lastly, we wanted to see whether these trends would change by year. So as you can see here, the total number of paper is increasing and has increased over the past 2 decades, but that is the case for most studies, so that is to be expected. And in particular, I would also like to draw your attention to how the percentage distribution in terms of percentage of different common spaces changes, with single subject space being extremely prevalent for the first, I would say, up until 2010, and then being gradually overtaken by off-the-shelf registration and templates. Therefore, we can appreciate a trend going from single subject space to atlas-based approach. And while this might have some benefits in terms of comparability of the study and standardization of the field of infant neuroimaging, some challenges still exist. And these challenges are quite unique to the age range that we're considering because of the great amount of change and brain growth that happens in the first years of life. As the next step, we're now investigating what the interaction between categories might be. So for instance, we know that earlier studies are more likely to use single subject space, as well as preterm studies of recruiting preterm are more likely to use single subject space. So we would like to investigate this interaction to assess what the effects might be in terms of distribution of common spaces. I would like to thank my lab, the Minds Lab, in particular Dr. Shinas, Dr. Dufford, Hannah Peterson, Alice Han, and Saloni Mera, who helped me collect the data and analyze it. And also I would like to remind everyone that Alice is also presenting a paper on this research and a poster on this research, and it's poster 3099. And I look forward to hearing your questions in the live session. Thank you.
Hello, my name is Alex Dufford, and today I will talk to you about a study called Low Infant Functional Connectome-Based Identification Accuracy Across the First Year of Life. People are unique and have individual variability. It's not surprising then that there is individual variability in the brain. However, the individual variability of the brain is only beginning to be understood. It is critical to understand how this individual variability may underlie typical and atypical brain development. Studies have demonstrated uniqueness and stability in both functional connectivity patterns and functional networks. However, it's currently unclear how and when these patterns of uniqueness and stability develop. The goal of this study was to examine functional connectome stability and uniqueness in the first year of life. The first year of life is also a developmental period of rapid and unparalleled brain development. Therefore, it's critical to understand individual variability in the functional connectome in this sensitive window. To examine the uniqueness and stability of the functional connectome in infancy, we use functional connectivity to measure the functional architecture across the whole brain. Functional connectivity measures the correlation in blood oxygenation level dependent signal as measured by fMRI, and its goal is to find synchrony between brain regions. Regions with high functional connectivity have high correlations in both signal. We can examine functional connectivity across the whole brain using functional connectomes, which are comprehensive maps of functional connections in the brain. Functional connectomes can be represented by a correlation or connectivity matrix in which nodes refer to the individual brain regions of a parcellation and edges refer to the correlation value between nodes. Edges can be thought of as a measure of functional connectivity between two regions. Spann et al. Demonstrated the uniqueness in functional connectomes by developing the connectome-based identification procedure, also known as fingerprinting. Through an iterative process, each participant from a different session, session 1 or session 2, is denoted as the target. The Pearson correlation between the target and all their matrices in the database is computed. A correct identification occurs when the highest Pearson correlation coefficient is between the target in one session and the same individual in the second session. This process is repeated until each participant served as the target once and is repeated for all participants, sessions, and database target combinations. The connectome-based identification procedure results in 2 ID rates, reflecting the 2 possible configurations obtained by exchanging the roles of target and database sessions. ID rates are computed as the percentage of participants whose identity was correctly predicted out of the total number of participants. Using the Human Connectome Project data, Finn et al. Showed high identification rates across resting state and task functional connectomes. The frontal parietal network emerged as a network that highly contributed to successful identification. Two studies have found evidence of the stability of the individual functional connectome in adolescence. Horan et al. Found that connectomes were stable across periods of 3 months to 1 to 2 years. Another study found high identification rates in adolescents 1.5 years apart. However, it is unclear how and when this uniqueness and stability develops. This study examined connectome-based identification across the first year of life. To examine this question, the current study used both an infant and adult sample. The infant sample was downloaded from NDA, and infants were scanned during their natural sleep using fMRI at 1.5 months and again at 9 months of age. We conducted connectome-based ID for both resting state and a passive listening task. We used this infant-specific functional parcellation to calculate connectomes, and the amount of usable data depended on the analysis between 15 and 47 infants. To compare with adult connectome-based identification, we used the resting state data from the Human Connectome Project and used the same infant-specific parcellation in the adults in order to draw direct comparisons. We found that the individual infant functional connectomes are not unique. Within ID rates were consistently the highest, between 48 and 70%, with lower ID rates for between session analysis. Between 14 and 26%, so very low. For adults, however, we found that the ID rates were much higher at 71.6% with the infant atlas, and this is shown to be above 95% with the HCP data for an adult atlas. As with previous connectome-based ID studies, we are interested in which brain edges across the brain contributed most to successful identification. In the current study, we're interested in if these edges would be different based upon development, infant versus adult. We calculated differential power, or DP, across the functional connectomes, which is calculated as the product of the edge value from session 1 and session 2 from the same participant, and is compared to the product of the same edge value from session 1 and session 2 from unmatched participants. If the within participant product is higher than the between participant product across all of the participants in the sample, the edge contributes highly to a successful identification and is indicated by a high DP value. For DP, we found that the number of connections within and between the prefrontal cortex were not significantly different and showed strikingly similar patterns. However, we found that the frontal connections in infants were significantly weaker. We also examined retest reliability for the infants and compared it to adults. We found that infants have lower retest reliability across resting state and task. Further, we found that edge level interclass correlation coefficients between the infant and adult edges were positively correlated. This suggests that while infants have much lower ICC, the same edges across infants and adults have high ICC. We were also interested in examining if low connectome-based ID rate in the infants was attributable to lower retest reliability. However, we did not find an association between ICCs for infants and adults. This suggests that the low connectome-based ID rates in infancy is not driven by lower retest reliability and supports the hypothesis that the low ID rates were attributable to differences in brain functional development. In this study, we demonstrated that compared to adults and adolescents, connectome-based ID rates are low in the first year of life. We found that similar edges, including prefrontal edges, contribute to successful ID in both infants and adults. However, we found prefrontal connections in the infants, which were much weaker, which likely explains the ID differences, as the prefrontal connections have been shown to contribute highly to successful identification in adults. We also found that retest reliability of edges was lower in infancy, but was not correlated with ID rate. These findings suggest that in the first year of life, the brain scaffolding is in place, but frontal edges are not quite developed yet. Future studies will be needed to examine when adult-like connectome-based ID rates are established developmentally. I'd like to thank the support of the Minds Lab at Yale School of Medicine, especially my mentor, Dr. Dustin Shinos, and the study's co-authors, Stephanie Noble and Siwan Gao. This study is currently available as a preprint on bioRxives, and links to the data and code can be found below, and I'd enjoy to hear any of your thoughts and questions on the study. Thanks.
Hello, my name is Lucille Moore, and today I'll be talking about generating individualized resting state networks in infants with template matching. Over the last several decades, functional MRI has revealed that the brain is organized into a set of networks that integrate to support behavior and cognition. And when communication within or between these networks is compromised, it results in poor cognitive performance and atypical behaviors. Resting state networks are functional networks comprised of regions that are cyclically co-activated at rest. So when you correlate the activity of functionally significant subregions from resting state data, you can see larger areas of similar connectivity belonging to the same network start to pop out. Multiple studies in recent years have utilized functional MRI in combination with graph theory and machine learning to identify and map a fairly consistent set of 12 to 14 resting state networks. In general, the majority of resting state networks observed in adults have also been identified in infants in some form. For example, here is the default mode network shown for neonates, 1 and 2-year-olds, and adults. But there is certainly variation in the specific network topology at different developmental stages, which are thought to reflect increasing integration within networks and segregation between networks over the course of development. Different networks also have different developmental timelines. So the first networks to come online are the primary sensory motor and auditory networks, followed by the visual, attention, and default mode, and finally followed by executive control networks. There is interest in identifying resting state networks in infants, in part because at younger ages, it's difficult to reliably measure higher order cognitive skills from behavioral assays and task-based MRI. Resting state fMRI makes it possible to examine the development of relevant functional brain networks beginning at birth. And this potentially allows us to identify individual differences at a very early stage that may be associated with, for example, higher risk of developing psychiatric disorders later in life. Another layer of complexity is the fact that the precise network topology of these systems has recently been shown to be highly specific to individuals in adults. For instance, in Gordon's 2017 Neuron paper, one thing that they show is that networks identified from task data can also be identified from resting state data, which gives us confidence that resting state networks truly represent functionally significant connectivity in the brain. And in addition to this, they also showed that the topology of networks derived from task versus rest data are very closely aligned within individuals. And when they compared the alignment of task and rest networks between different subjects, the regions were clearly not as well aligned, suggesting that network topology is indeed highly specific to individuals. And unfortunately, these individual network distinctions are lost in most studies due to averaging, which may weaken measures of brain behavior associations. The specificity of networks in infants is an open question, and answering this question is difficult due to the large amount of rest typically required to identify individual networks. So for instance, the Gordon study, each subject had about 5 hours of rest. And this is difficult enough to collect in adults, but even more difficult in infants for several reasons. One of which is that infants must be sleeping, which decreases the signal to noise ratio of the data. They tend to move a lot, introducing motion artifacts into the data. And in general, infant data is difficult to process due to the inherently lower data quality. Our group is exploring the possibility of using a community detection method known as template matching for use in infants. Compared to approaches that are purely data-driven, template matching generates individual network maps by comparing the whole brain connectivity of a single subject to a basic expected network structure. This approach is advantageous for infants because it reduces the noise inherent to purely data-driven approaches and therefore requires less data to identify networks. In order to use this technique in infants, I first had to generate an infant template. For this, I used 69 neonates from this UCI study with more than 4.2 minutes of rest for each subject. And we were able to identify 12 resting state networks matching those observed in adults. So here are the resulting templates for each network, where the warmer colors reflect voxels that are positively correlated with the average signal of a network. And the cooler colors are voxels with negative signal correlation. While we observed all major adult networks and the patterns of connectivity look generally similar, particularly for networks that are known to come online earlier, such as the auditory network. There are certainly some interesting differences that I don't have time to get into today, but for example, one thing that really popped out was that in infants, unlike adolescents, the motor cortex is very negatively correlated with other areas in the majority of the observed networks. To use the template to define individualized network assignments for a single subject, you first generate whole brain connectivity matrices where you correlate the bold signal of each voxel against the bold signal of every other voxel. So this image, for example, is showing the whole brain connectivity of a single voxel in the posterior cingulate cortex. And then for each voxel, in order to assign it to a network, we use eta squared to identify which network template has the most similar whole brain connectivity patterns. Each voxel is then assigned to whichever network its connectivity is most similar to, resulting in individualized network maps for each subject, where each voxel is assigned to one of the 12 resting state networks. Here are some examples of individual networks generated in 4 UCI subjects. And the networks look pretty reasonable, even though they were only generated using 4 to 6 minutes of data. How do we know that the networks generated from template matching are truly specific to individual infants? The majority of the work I've done to date has focused on this question, and we plan to interrogate it using multiple methods, but today I'll mostly focus on split halves reliability analysis. So for this analysis, we take subjects with a large amount of data and compare the similarity of networks generated from the first versus second half of the subject's time series data. And you would expect that if the technique is robust, the networks generated from the same subject should be more similar than networks generated from different subjects. We were very fortunate to be able to test the validity of the approach in 2 independent neonatal data sets, one called Exodo and the other one from WashU, with more extended acquisition times that are typical for infants. So after selecting subjects with at least 20 minutes of resting state data, the Exodo data set had 14 subjects with an average of 39 minutes per subject. And the WashU dataset had 26 subjects with an average of 32 minutes of rest for each subject. These are examples of individualized networks generated from the first and second half of a single subject's data across study groups, with adolescents at the top and the neonatal networks in the second and third rows. Just by eye, you can see that the network maps generated from each half look fairly similar to one another. And we quantified the similarity using mutual information. Here I'm showing the normalized mutual information on the y-axis when comparing networks generated from the same versus different subjects, shown in gray and black respectively. Dr. Marisa from our group has shown previously that in adolescents, the normalized mutual information from same subject networks is significantly higher than from different subjects. And though it's less discrete, the infant data set also shows this same positive shift in the same subject mutual information compared to the null, which is exciting because it suggests that template matching effectively generates networks specific to individual subjects in infants as well as adolescents using relatively little data. And we also noted that when comparing adolescents to infants, the same subject mutual information is pretty consistently high across groups. However, there is a significant positive shift in the null distribution in infants compared to adolescents. And this is super interesting because it suggests that infant brains are less differentiated from one another compared to adolescents, which very much fits with our understanding of development and resting state data representing a shared history of coactivation. An essential question for this technique is how many minutes of rest is required to generate stable networks? Again, Dr. Marisa from our group showed that template matching can generate reliable subject-specific network maps in adolescents using as little as 10 minutes of resting state data. And I need to analyze this question more carefully still, but for now, I'm simply graphed the same subject mutual information versus the average minutes of rest per half for each subject in both the Exido and WashU neonatal data sets. And there is a significant positive linear correlation for both groups, which suggests that even within this range of data, significant improvements can be made by adding just a bit more data. And to get an idea of network specificity, here I'm showing the 5th to 95th percentile of the null distribution for Exido. And you can see that it's not until around 20 minutes that points start falling well outside of this distribution. And the same can be seen for the other neonatal data set. So I still need to look at this more carefully with some other analyses, but it looks like infants likely require more than 20 minutes of resting state data to achieve high network specificity. There are a lot of other analyses I'm currently working on to further verify this approach in infants. But one of the main future directions is to compare the topology of task-evoked bold responses with individual networks derived from resting state data. So similar to what the Gordon paper did. So in WashU neonates, they performed an auditory oddball task paradigm that in neonates activates auditory cortex as well as other cortical areas that respond to deviant stimuli. And we plan to repeat the analysis done in Gordon's 2017 paper that I discussed before to see how well the topology of the auditory task activation aligns with the network maps I generated using template matching. So in conclusion, we were able to use template matching to identify a set of resting state networks similar to those identified in adults. And these networks can be generated using only 10 to 20 minutes of resting state data as opposed to hours. The infant networks are a bit different and they're less differentiated, which makes sense in the context of our understanding of neural development. And this work will be critical for future efforts to study the early emergence of individual functional network topology beginning soon after birth. Thank you so much to my mentors, Drs. Alice Graham and Damien Fair, all the people in our group who have contributed to this project, and of course, all of our collaborators who provided the neonatal data sets and input on this project. Thank you.
Hi, everyone. I'm really excited to present our work related to intergenerational pathways, the effects of maternal experience of discrimination on the offspring brain. My name is Marisa Spann. I'm at Columbia University. So first, what is racial discrimination? It's treating someone unfavorably because of his or her race or personal characteristics associated with race, such as skin color and facial features. This is considered a psychological stressor, and it's well established in adults that it can lead to increased mental health disorders such as depression and anxiety. What we don't know are the neural substrates that may contribute to the effects of discrimination. In discrimination during pregnancy, there's even less information, but similar to non-pregnant adults, there's an increase in psychiatric outcomes, as well as increase in physical outcomes such as systemic inflammation, as well as chronic health conditions such as gestational diabetes, and behavioral outcomes in terms of those related to the maternal experience of medical professionals, which could lead to prenatal care delay, and then ultimately disruptions in health outcomes. So we have to turn to literature related to neural substates of discrimination in adults, non-pregnant adults, in part because there really is a paucity of literature in this area. So what we do know is that in samples of predominantly Black adults, there's been findings of greater social discrimination associations with higher intrinsic connectivity of the amygdala, which is involved in limbic circuitry, as well as increased connectivity of the amygdala to other brain regions involved in emotion regulation, as well as more subcortical areas related to inhibition. Associations also have been identified as independent of psychological factors. And so this is really unique to discrimination. And also turn to literature related to race face processing, again, because it's really a single study, as I mentioned, that's really looked at brain associations with discrimination. And those studies that have looked at Black versus white racial face processing, and really the idea is in-group versus out-group face processing. So the other face. And what they've identified is similarly amygdala connectivity and its association with brain regions involved in face processing, as well as they're denoting racial bias, but we traditionally think of some of these brain regions associated with executive functions and emotion regulation as well. And these findings were not necessarily consistent in terms of the direction of connectivity, so we won't sort of really be focusing on that here today, but just to know sort of broadly what the brain regions are that have been identified. So when we think about intergenerational transmission, what we're thinking about is the pregnant woman's experience for the generation before experience and how that relates to the offspring, so the next generation's behavior, brain systems, and ultimately quality of life. And so we sought to understand how the pregnant woman's experience of discrimination translated to the developing fetal brain. And we proposed, given the existing literature, that higher maternal discrimination would be associated with weaker newborn connectivity in brain regions associated with racial processing and experience of discrimination. So thinking about amygdala connectivity, as well as some of the cortical and cortical brain regions identified in previous studies, excuse me, such as the prefrontal cortex and hippocampus. So this was a sample of 45 pregnant adolescents, age 14 to 19, who underwent routine prenatal care. This is a predominantly Latinx sample, and they came in the second or third trimester. They completed the experience of discrimination scale. They had fetal behavioral assessments, as well as biospecimen collection. And then within the newborn period, the infants came in for an MRI scan, and there was subsequent follow-up assessments of behavior. For the larger study, this was an R01 funded through NIMH, and the PIs of that R01 were Dr. Catherine Monk and Bradley Peterson. The pleasure of using the data. And so you can see here, in terms of demographics, the sample is around 17 years in terms of mean age, as well as education. You can see that majority of them are in the around the 12th grade, 12th grade level. And then really another half is within high school age, as we mentioned, predominantly Latinx sample. And the main take home really here is that the sample is pretty healthy if you look at gestational age of birth. It's not a preterm sample, and the birth weight, APGAR scores are all within typical age range. And so, as I mentioned, we performed an MRI with these newborns. I think that having an infant-specific MRI is pretty cool. At some point, I'd love for our location to get that. And I don't have to tell you all what resting state functional connectivity is, but just to remind you that we have 2 seed areas that we were particularly interested in. I guess it's a total of where we have the so 4. So it's bilateral amygdala and hippocampus. And we're looking at connectivity between those seed regions and the whole brain. And so the main findings, we identified that high maternal experience of discrimination during pregnancy was associated with decreased connectivity of the right hippocampus to the dorsolateral prefrontal cortex. The findings with the left hippocampus were non-significant. And then for the left amygdala, we found that higher maternal experience of discrimination during pregnancy was associated with reduced connectivity again between now the left amygdala and the interior portion of the prefrontal cortex. And again, there was non-significant findings with the other hemisphere, so the right amygdala. Just to recap, so this would be the first study to look at the intergenerational transmission of the effects of discrimination on the newborn brain. And that maternal experience of discrimination was associated with decreased connectivity of the hippocampus and amygdala to aspects of the prefrontal cortex. This is pretty consistent with those few previous studies that were either looking at experience of discrimination or race-based processing. The other race in adults. And so next steps, we're looking at the interaction between maternal stress and acculturation. We're looking at the relationship between discrimination and birth outcomes and fetal growth. And we're also particularly think they would be interested to understand the development of processing racial differences in conjunction with the intergenerational model. So considering how mom's experience and perceptions then translate to the development of her offspring in infancy, as well as how this then relates to emotion regulation and attention in the dyad. So I'd like to thank my collaborators, again, Dr. Peterson and Monk, and the research participants and the funding agencies. Obviously, this work could not happen without you. And I'd like to thank all of you for joining this, coming to see this talk, and looking forward to seeing you at the meeting. Thanks.